Hello again, biology, and welcome back. We're going to start chapter 7 today. This one's on cellular respiration. Cellular respiration is I like to think of it as almost the exact opposite of what we just learned with photosynthesis. With photosynthesis, we take energy, and we take water, and we take carbon dioxide, and we create glucose and oxygen. And on the flip side, for us animals and for plants who have mitochondria, any organism that has mitochondria, we're going to take glucose and oxygen, and we're going to burn glucose essentially into carbon dioxide, water, and create energy, ATP, and heat. So let's jump right into it. All right, so let's jump into and define what cellular respiration is. It is the process in which cells make adenosine triphosphate, or ATP. We've talked about ATP before. This is the energy molecule that organisms use in their cells. Plants are able to create some ATP. They're able to store energy in glucose. So plants can make energy and then burn that energy, use it all winter if they lose their leaves. But both autotrophs and heterotrophs use cellular respiration to break chemicals such as glucose down into simpler molecules and to acquire heat from them and energy. So there are two parts to cellular respiration, and we're going to go over the first one, which is glycolysis, today, and then I'm going to post the second one, aerobic respiration, shortly after this. But in glycolysis, we're going to take organic molecules such as glucose, and we're going to break them down into a 3-carbon molecule we call pyruvic acid. Pyruvic acid is the product of glycolysis, and it's important. It's the one that it's got a lot of energy trapped inside of it, and it's the one that our cells are really, really good at extracting the energy from. The process of glycolysis does not require oxygen. So we think that glycolysis is one of the oldest methods of acquiring energy. We think bacteria were able to do it long before there's oxygen in the atmosphere. The second part of this, and the one that makes eukaryotes kind of unique, is we're able to do aerobic respiration. For this, we need oxygen, but we're able to break down pyruvic acid further to form ATP. In glycolysis, we just get pyruvic acid. In aerobic respiration, we take that pyruvic acid and we break it down. There are some alternative pathways to glycolysis. So once pyruvate is created, it can go several different ways. It can either go through aerobic oxidation, and that's what we're going to talk about later with the citric acid cycle or the Krebs cycle. But the other two, these fermentation pathways, are what happens if there's no oxygen, and it can either have alcoholic fermentation or lactic acid fermentation. All right, the equation for cellular respiration is as follows. It is C6H12O6, or glucose here, glucose, plus 6O2, which is oxygen, and this goes to, and these reactants yield these products: 6 carbon dioxide, 6 water, and 38 ATP. And we will figure out how to tell you exactly where each of those ATP are coming from. All right, so there's a little extra credit assignment hidden in here. You need to write the equation on the last slide on a note card, show it to Mr. Greer, hold on to it. If I'm walking around and I ask you the equation for cellular respiration and you can give it to me, I'll give you extra credit on your quiz. So just a little tidbit hidden in here. All right, so let's jump into glycolysis, this first part of cellular respiration. And this is a pathway in which we're going to take one 6-carbon molecule of glucose and we're going to oxidize it down into 2 3-carbon molecules of pyruvic acid. Now, this is important. We're not losing any carbon molecules. We're just making 2 3-carbon molecules. Add those together and you still have glucose. Glycolysis takes place in the cytosol, or that cellular fluid outside of the mitochondria. So, and it happens here in all organisms. So bacteria can produce glycolysis because they have a cytosol. Eukaryotes can, plant cells can, fungus can, all of them. There are 4 steps to glycolysis. We're going to kind of go through them one by one. The first is 2 phosphate groups are added to 1 molecule of glucose. So this is a molecule of glucose over here. It's important to note that this glucose has 6 carbons. We add these phosphates, which I have put in red and put a P on because they're not carbons like these. And we're going to take 2 ATP and we're going to break them down to 2 ADP. And ADP is adenosine diphosphate, meaning 2 phosphates. So each one of these ATP gave up a phosphate molecule to put it on either end of this glucose. All right, now we're going to take this 6-carbon molecule that has the phosphates on it, and we're going to split it right down the middle into 2 symmetrical molecules. We're going to turn it into 2 G3P molecules. And you don't have to write this down, but this one should be familiar. You've seen G3P before. You saw it in the Calvin cycle with plants. The processes are surprisingly similar frontwards, backwards. But now we have this 2 of these G3Ps here, and we're going to send those on to the next stage. At this point in time, we're going to take each G3P and we're going to oxidize them, and they're going to receive a phosphate group. So remember that every time that you have an oxidation, you have a reduction. This is going to be important later. This NAD plus here is going to turn into NAD, NADH. Now, the reason that we're putting all of these phosphates onto this molecule is each time we put a phosphate onto this molecule, we add energy to it. Phosphates are very high energy molecules. By adding them on, later we're going to strip them off, and we're going to get a lot of energy from that. But the important thing here now is we've taken this G3P and we've turned it into having 2 phosphate groups again on each molecule. And we've created this 2-NADH, this high energy electron carrier here. That's now, we're going to talk about why we need to get it back eventually. All right, on this last stage here, step 4, we're going to take the phosphate groups off of the 3 carbons. And now we have pyruvic acid. These new 3-carbon molecules are pyruvic acid. Each phosphate is going to be combined with a molecule of ADP, or this adenosine diphosphate, to form ATP, adenosine triphosphate. So glycolysis makes us 4 molecules of ATP at the end. Now, it's important to note that in this process, here's the diagram for this 4 ADP turning into 4 ATP here and releasing some water. But it's important to note here that we had to put in 2 ATP at the beginning to put phosphates on that glucose. So we had to use 2 ATP and we created 4 ATP. That means that we actually made 2 brand new ATP, and we recycled 2 ATP. So glycolysis creates 2 ATP for every glucose. This is important. We're going to talk about why it's way better to use all of cellular respiration instead of just glycolysis, but this is a good start. All right, now we can talk about fermentation. Without oxygen, you can't have cellular respiration. We don't go into the Krebs cycle, citric acid cycle. So we need to take this NADH and be able to use it again. So what we do is we have to convert it back to this NAD plus to continue glycolysis. So we can reuse it and keep going through glycolysis and keep getting those 2 extra ATP. Anytime that we have a process that's regenerating, we call it regenerating this NAD plus. Without oxygen, it's called fermentation. And fermentation does not produce any energy. It just sets glycolysis up to start again, to use more glucose. So, no ATP produced during glycolysis. All right, in lactic acid fermentation, we're taking enzyme and we're taking this pyruvic acid here, and then we're going to turn it into a 3-carbon compound called lactic acid. When we do this, we're going to transfer this H here to this pyruvic acid, and 1 free proton. What this is going to do is this is going to turn this into 2 NAD+. And so now we have this 2 NAD+ again. It's able to go back up and start glycolysis again. It's able to take this glucose, turn it into pyruvate. Each time that this happens, it makes 2 new ATP, and this process continues over and over and over again. This fermentation here just takes this NADH here and makes it usable again. Lactic acid has a lot of uses. It's used by a lot of organisms to get their energy. When milk spoils, it's because of those organisms that are inside of milk using lactic acid fermentation to create energy. This creates things to make cheese, buttermilk, yogurt, and other dairy products. I mean, lactic acid fermentation can't be that bad. Look at all these delicious cheeses here. Your muscle cells also use lactic acid fermentation during strenuous non-aerobic respiration. So when your cells stop getting oxygen, they start using lactic acid fermentation to get a little bit of energy. And this accumulation of lactic energy reduces the muscle cells' ability to contract, and it causes them to cramp, causes pain, and it causes fatigue. So whenever you get those muscle fatigues, it's because you have this lactic acid fermentation going on in your cells. That's why stretching is really good. It helps to push that lactic acid outside the cells and helps the body eliminate it. All right, now we're going to talk about alcoholic fermentation. Plants and unicellular organisms like yeast use this to create, to get their glucose back, get their ability to do glycolysis back. And in this, they're going to take pyruvic acid and they're going to turn it into ethyl alcohol or ethanol. And it takes 2 steps to do this. They have to do a decarboxylation, and then they have to regenerate that NADH. You don't have to know the names of them. We're just going to talk about the process real quick. So the steps to alcoholic fermentation. The first is we create a CO2 molecule per pyruvic acid, so 2 for 1 whole molecule of glucose. And we remove it here to make a 2-carbon compound. This 2-acetaldehyde here, it has 2 carbons. So we've taken this 3-carbon pyruvic acid and we've turned it into a 2-carbon molecule. We're then going to take 2 hydrogen atoms and add it to this to create, one goes here and one goes here, to create this ethanol now, or this ethyl alcohol. We get this NAD+ again, so now we're able to take that, go up and do glycolysis, move glucose to pyruvic acid. We create 2 ATP each time we do this. So in the same way as lactic acid fermentation, we're regenerating and continuing the cycle so that these cells can continue to get energy. CO2 is released. This is important in bread production. If you've ever made bread and you've opened it up and you see all the little bubbles on there, that's all the yeast doing alcoholic fermentation to make this carbon dioxide. So pretty cool little science you can see thing going on there. The last thing we're going to talk about in 7.1 for today is the efficiency of glycolysis. Here's some yeast. I wanted to show you guys what they look like. These are the little guys that help make your bread. Thank you, yeast. We measure energy in the kilocalorie, and so what that means is 1 calorie equals 1,000 calories, and most of the time the stuff that you see on like your nutritional labels is in kilocalories already. But releasing all of the energy in glucose creates 686 kilocalories of energy. So if we took that pyruvic acid and we had oxygen and we used cellular respiration and did the whole thing, we would get 686 kilocalories of energy. Release of energy from glycolysis only creates 14 kilocalories of energy. Glycolysis is not very energy effective. It would not be enough to sustain large organisms like humans, like anything bigger than like a bacteria or a protist. So early bacteria were able to survive using just glycolysis and fermentation. They were able to regenerate that. They were able to use other food sources. They were able to carve out a place for themselves on the planet. But for us, we're going to need aerobic respiration, which we're going to talk about next. But this is the end of 7.1. Thank you for watching. See you in the next one.